Today's lab we're going to talk about soils and study some of their physical properties and how they relate to environmental problems and issues. Soils are a real vital and important resource on the surface of the earth because they're important for sustaining life as we know it, animal and plant life, and also they just add to the overall quality of the environment. They're an important source of the cycling of elements such as carbon, oxygen, nitrogen, and phosphorus between the earth and animal and plant life. And they also serve as an important filter for any kind of groundwater. Any water that hits the surface of the earth must first infiltrate through the soil zone. And so whatever is on the surface of the earth will get ultimately carried into the groundwater system. So if we start polluting the ground or the surface of the soil surfaces, we're going to carry those pollutants into the subsurface and ultimately maybe into our drinking or usable water. Because of the importance of soils, we're going to try to understand some of their physical properties, because their physical properties are really what controls how both nutrients are held or leached, and also how pollutants are held or leached. So it's really important to understand their physical properties. Pollutants are added to soil surfaces either through atmospheric deposition, say from ash or smoke from industries, mercury sometimes from burning coal, that sort of thing. They can also gain pollutants from just the physical dumping of materials on the surface, such as, every time farmers add fertilizer or herbicides or pesticides, we're actually adding something to the surface. They can be contaminated by animal waste on farms, for example. And in urban settings, such as Newark and any place that's developed, we can add pollutants through dumping of residues from manufacturing. And this has happened commonly in various parts of New Jersey. Now, soils in natural setting are essentially formed in place. They are formed by the physical and chemical breakdown of the parent material, which might be bedrock, it might be other sediments, or it could even be other soils. And what we call this breakdown is weathering. Now, soils, again, form essentially in place, and there's 6 things that contribute to the forming of soils. The parent material, what kind of stuff was underneath, where the soil is forming, whether it's on a hillside or in a valley. Also, what kind of organic matter is growing on the soil. Are it trees, is it grasses? Again, how much precipitation there is it really wet, is it really dry? You obviously will form soils more quickly in a very wet climate versus a dry climate. And also time. It takes a long time to form a soil. 2 or 3 foot soil forming in our neck of the woods takes about 15,000 years to form. So anything we do to the surface, if we take away soil, if we pollute soil, it won't get replaced in any reasonable amount of time. So I'm going to draw a couple of diagrams on the blackboard of a young soil and an old soil so you can see how soils develop over time. First, let me show you just a simple profile of a fairly young soil. We have the ground surface. There might be a really thin organic matter layer, which I'll call an O. And then underneath here will be rock, which is unaltered bedrock. Could be loose sediments as well, but it's essentially unaltered and unweathered. And then in between, there's going to be kind of a kind of fuzzy dividing zone, where in this upper part, we start to have an A horizon develop, which is where we're going to have weathering starting. But this is typically a zone of leaching. And what happens here is that the original parent materials that are present in this bedrock are slowly but surely affected by the infiltration of rainwater. And we start to break down those minerals kind of in the inverse of Bowen's reaction series. But nevertheless, they break down fairly slowly, and it does occur. And the minerals are breaking down. They release solutes or little dissolved ions. And those ions are basically trickling downwards, both in the water and also physical particles. During the weathering of these materials, you start to make smaller and smaller and smaller minerals. And these minerals might be clay minerals, which are like sheet silicates, very fine-grained sheet silicates, like very fine-grained mica is very similar. Or they may be what we call just a general aluminum or iron oxides. And these materials, we'll find out in a minute, are very, very fine-grained, very much like flour. And they're also physically carried downward along with the water. And what's underneath this zone is kind of a zone we'll just call weathered bedrock. And it's going to have big pieces or chunks of the bedrock, but any zones or joints, you're going to start having these weathering effects occurring along any zones or cracks or joints. Now this would be a representative of a fairly young soil just starting to develop, maybe something that's less than several thousands of years old. Or this might also represent a soil that could be in a very arid climate where there isn't a lot of rain going on. But in our area, where there is a lot of moisture, you tend to develop soils with a little bit more character to them. And I'll draw a picture over here. Again, we'll have this organic matter at the top surface. This would be our O layer. But instead of having just this A layer, which is, again, the zone of leaching, we end up having another layer kind of developing above our altered bedrock and above our rock. Now, I should mention too that this is not what's happening as this goes on in time, is that the surface of the ground doesn't really change, but the zone that's weathered gets deeper and deeper and deeper. So what happens here now is that from this zone of leaching, of all these little tiny clay particles and solutes and so on and so forth, the organic matter ends up, sitting in this zone. And this zone is oftentimes called the zone of accumulation. Now, how many of you have ever dug in your garden and dug a fairly big hole to plant a tree, and you've actually seen a difference in color between the upper surface soil and this deeper soil? And usually what happens is this upper soil tends to be more black, it has more organic matter in it, and typically this lower soil tends to have more yellowish color or reddish color, and that's because it's accumulating all these iron and aluminum oxides that tend to be kind of rusty red, rusty yellow, or kind of whitish. And so there is a definite difference that develops over time. This is a soil profile. Now we're going to describe some of the soil properties and how they might affect the migration of fluids or pollutants. The first thing is that the physical and chemical weathering of the parent material is going to break down the original minerals into finer grained particles. And as the soil ages with time, generally the particles end up getting smaller and smaller and smaller. And again, the things like olivine and pyroxene are going to decay into clay minerals and some of these iron oxides. And things like feldspars and micas and quartz may ultimately end up also as clay minerals and tend to be more aluminum oxides. Organic matter is also added to the surface. So we actually measure these soil properties in the laboratory, and they help to interpret the processes that form the soil, and also something about how the water might pass through soil, and also something about the parent material. The first thing that's probably the easiest to identify in a soil is its color. Now, color tends to reflect some of the soil composition. Obviously, if you have a very black or brown soil, that has a lot of organic matter in it, and it tends to be dark colored. If you have something that's very light tan or light gray soil, it's very organic poor and be made up primarily of minerals. Yellow or red soils generally contain these aluminous and iron oxides. The next physical property that we're going to deal with is texture. And again, the texture of a soil is again related to the grain size of the soil and also a little bit to its chemical properties, but mainly grain size. We basically divide the soils into the three main groups: sand and silt and clay. Now, when we deal with the soil textures, most people that deal with soils determine the amount of clay, sand, and silt by sieving and doing various other settling techniques to determine how much is there, and then they plot the composition on a ternary diagram. And at each corner of the ternary diagram represents 100% of that material. And you can see a bunch of names in this ternary diagram. And kind of at the bottom center is this name called loam, that has about 10 to 30% clay in it and about equal parts of sand and silt. And it turns out that this composition has just enough of the right physical properties that it is a really good soil for growing. So people strive to get some kind of loam in their gardens or soils. Now, what's important about knowing the grain size is that the grain size directly correlated to the surface area of the soil itself, which affects two things. It affects how materials are held to the particles in the soil, and also how water can move through the soil or its permeability. So first, the surface area. I have listed here the sand, silt, and clay, the grains, and their respective surface areas. Now, sand has about a surface area of 30 centimeters squared per gram, and a gram is about a quarter of a teaspoon. If you filled up a Coke about halfway full, that would be about the surface area on sand. So you can imagine that when you get to clays, and that's like 3 million centimeters squared per gram. Now, what's important about the surface area is that the more surface area you have, the more easily water or pollutants or nutrients can be attached kind of to that surface area. There's very small little forces that hold molecules to the edges of these grains, and if you have more edges and surface, you're going to have more of these attachments. Being able to attach molecules or other particles or release them is called cation exchange capacity. So sand that has a really low surface area is going to have a really low cation exchange capacity because it doesn't have many sites for things to be held on in the first place, and what's there isn't really going to move too much. On the other hand, clay has an extremely high cation exchange capacity relative to sand. And so there's a lot of surface area for molecules to attach. There's a lot of potential for molecules to be exchanged as water moves through a material. Soils that have this high cation exchange capacity and surface area are better able to absorb nutrients, or they can hold water. And so on the one hand, they're very good for agricultural purposes, but on the other hand, they can also more easily contain or hold contaminants, or they can slowly leach out contaminants over a long period of time. So sometimes, soils that are contaminated, and if they are relatively fine grained, they can almost be considered like ticking time bombs for slow release of materials. On the other hand, you don't want too much clay because, the problem that you have with sand, silt, and clay is, again, the permeability. If you have too much clay, all the little clay grains are very fine grained and they all line up kind of on top of one another, and it's very difficult for water to trickle through, which is why farmers don't want too much clay in their sandy loams or their loams. On the other hand, if you have completely sandy soils, the water will go in so fast that there's no chance to really, it very readily leaches material, and there's no chance for other ions to be replaced. So there's a kind of a trade-off between how much clay, how much silt, and how much sand you want, and that's why farmers like to have that loam, which doesn't have too much clay, but just enough to hold all those nutrients that they need, and yet allows enough water to go through it so that it doesn't get waterlogged and soggy. This table actually summarizes a little bit better just the kind of properties of permeability. Again, sandy, silty, and clay soils, the permeability for sandy soils is going to be high and clay is obviously low. And to make a corollary of that, we're talking about the infiltration and the flow rate through the soil. So again, infiltration is going to be, and flow is going to be very high in sandy soils and relatively slow in clay soils. On the other hand, if you want to hold some water so that the plants can use it eventually, you want to have a little bit of this material. Obviously, a really sandy soil, the water is going to flow through really fast. There's not going to be very much storage available. A clay soil, it will hold a lot of that water. The amount of sand, silt, and clay are going to affect how water travels through the soil. Okay, the next important physical property of soils is the amount of organic matter that's contained in the soil. Again, plant residues that are deposited on the soil break down at the surface, typically by the reaction with oxygen, sometimes with an enzymatic reaction with microbes, bacteria, and fungi in the subsurface. And basically they form CO2, water, some energy for the bacteria to grow, and this residue that can't really be broken down as well, called humus. Now humus, not to be confused with the hummus that we love, is residual organic matter that isn't easily broken down. It doesn't look like any physical plant or twig or anything like that. It's just this sort of nondescript black gunky stuff that's in soils that you see. If you can identify a leaf pattern or a twig, it's still not humus yet. Now, humus or organic matter is also very important in the permeability, water storage capability, and cation exchange properties. And that's kind of summarized in the next table. Again, if you have a high amount of humus, you're going to be able to hold a lot of water. And again, you'll have a lot of absorption of nutrients or contaminants. So on the one hand, it's good to have a lot of organic matter if you want to hold nutrients in a garden, but it's not so good if you're trying to remove pollutants from a soil. And we're going to do a test in the lab, and I have actually the little key here about what color we're going to do. What we're going to do is actually take our soil and leach it with a solute that's going to kind of extract a little bit of that organic matter. And when that if that extract is really dark, amber yellow, kind of like recycled beer, I call it, then you know you've got a high humus content. But if it's really pale or yellow or colorless, then the humus content is very low. I should mention too that mainly the reason that gardeners want to add organic matter to soils when you commonly are doing a garden or something is you're trying to add both this water retention capability and also this ability to release some of the nutrients that are stored in the organic matter. The last physical property that we're going to measure is the pH of a soil. Now, I mentioned that a soil has kind of a surface and there's molecules and stuff stuck on the outside of it. And those molecules, that could be stuck on the outside could either be some of these, nutrients, minerals like sodium, potassium, magnesium, some of the things that are being taken away from the minerals, or they can be what we call hydrogen ions. If there are a lot of hydrogen ions, that makes the soil very acidic. On the other hand, if there are a lot of other ions like the calcium, magnesium, potassium, that makes the soil much more basic. And if you remember perhaps from high school chemistry, we measure the amount of acid and base using a pH scale. And if the pH is from 1 to 7, it's acidic. If it's 7 exactly, it's neutral. And if it's 7 to 14, it's basic. If you have these elements on the outside of the soil, those are important elements for the growth of plants. Hydrogen ions, on the other hand, are not so conducive to plant growth. And these soils that are very acidy, you'll commonly end up seeing problems with plant growth. And this is one of the problems that comes up with acid rain. If you have so much acid rain falling on a surface, you end up replacing a lot of those cations that could be used for plant growth with hydrogen ions. It makes it very acidic and plants will die. So those are the things that we're going to try to test for in the lab. We're going to test for texture and try to understand something about sand, silt, and clay. We're going to test for humus content. We're going to test for pH, and we're going to examine some of the colors of soils from three profiles that were taken in New Jersey. And some of these were on old soils, some of these were on young soils, and we're going to investigate kind of the differences between them. The first part of the lab is going to be just classifying the soil in terms of sand, silt, and clay. And what we have here are two examples of some soils from New Jersey, one of which has got some very fine grain material and just a few coarser grain materials, and one of which is mostly coarser grain material. And what we want you to try to do is look under the microscope, a binocular microscope, and we're going to tell you the proportion of clay that's present in both. And then we want you to try to estimate really how much sand is there, and we'll assume that the silt is the rest of the material to make up 100%. And how you're going to estimate how much sand is there, you're going to do two things with that. We have beside each microscope, we have a little chart that shows kind of how you would guesstimate how much percent of material is in each one, where this is like virtually 1% and this is like 50%. So you're just going to kind of estimate, look down the microscope and see which one kind of fits with the total material. And the other thing we're going to have by each microscope is a little scale that shows relative grain size, because we're going to ask you a few questions about the grain size of these things. The numbers that are, say, D equals are actually in terms of millimeters. Okay, so you can just look down the microscope, match up the sand size grains with one of these dots on here, and then you'll get the size of sand size grains. So we'll have a few questions on just estimating the proportions of the clay, sand, and silt. You're going to plot them on this ternary diagram, and we want you to then tell us what's the name of the soil. Is it a clayey loam, a sandy loam, silty loam, something like that. We want you to estimate the amount of sand in each of these, and we also want you to estimate roughly the range in grain size. And this would be a typical exercise that you might have to do if you're trying to understand the soil physical properties. This part of the lab is going to look at some of the soils in New Jersey. These were actually collected on the New Jersey surface. We're going to compare some old soils and some young soils, and some soils that were on different kinds of bedrock, and some soils that were either taken in glaciated areas or unglaciated areas. On page 7, there's a map that shows the location of the 4 different soil sites. And why don't we look at the bottom figure first? You can see that that shows a map of New Jersey covered by glaciers. And you can see that at Site 1, that was covered by glaciers. Site 3 was covered by glaciers. Site 2, however, was not. And Site 4, again, was not. And if you go up to the upper map, you can see what was the underlying bedrock in those areas that underneath the glacier itself. Well, at Site 1 and at Site 2, the underlying bedrock was actually these Watchung basalts. Site 3 is actually on top of some sandy and silty, kind of carbonate-rich shales. And again, that was the bedrock, but again, it was on a glaciated site. And Site 4 is essentially on some Ordovician limestones. So what we want you to just talk about first is in your lab, starting on page 9, there is this table that's going to summarize the properties of these different soils. And in fact, if you refer to that diagram, you'll see that actually at Site 1 we have actually two soils. These are from two different horizons in the soil profile. And at Site 2 we also have two different horizons in that soil profile, but only one each at 3 and 4. So the first thing to do is just, what is the underlying bedrock? What's the parent unit? And again, fill in the blanks as to what the parent unit was. And then we want you to put down, was that in a glaciated area or an unglaciated area of New Jersey? This will become important later when we ask some questions about the relative ages of these soils. Then we will start doing the actual physical properties. And the first one is going to be color. Okay, now in each laboratory in the front of the room, your instructor will have a tray with some of these soils in them that are moist. And in order to do the color of a soil, you always want to have the soil moist or wet. And so here we've got the soils 1A, 1B, 2A, 2B, and soils 3 and soil 4. And what you want to do is fill in your table, part 3 on the third column, just what is the color. And you can use sort of a blackish brown perhaps for this one, or blackish gray. This would be kind of a red-brown. This is kind of just a brown, that sort of thing, okay? And just go ahead and just use your best guess as to what the color is. Okay, the next column that you're going to fill in has to do with the texture of the soils. And the way we're going to do this is the soils are up at the front of the room, and what you do is simply take a little bit of the soil. This one's a good example because it's kind of fine grain. And you put a little bit between your fingers, and you can also look at the soil itself in the cup, but also based on what you saw in the first part of the exercise, you're going to roll a little bit of this between your fingers. And obviously if you can see sandy grains or feel sandy grains, it's going to feel real gritty. But if you end up with a residue on your finger, like I have this kind of yellowish residue on this, chances are there's quite a bit of clay in there. But what we want to do is just get approximate. If it tends to be more clayey or more sandy, why don't we go with those two extremes? So you could call it something that's sandy or clayey loam. All of these are basically loam samples. So we're just really trying to go for, is it tends to be more clayey or does it tend to be more sandy? Okay, the next part of the lab is going to test the actual pH of the soils that we've collected. And to do this, what we're going to do is take a little bit of soil, put it in one of these little containers like you did for the color, and we have this test kit that's actually produced by the Lamont Chemical Company. And this is a kit that you might use out in the field if you were just trying to do a survey while you were actually collecting the soils, so you'd have an idea of what was happening with the soils. Now the first part of the test we've already actually done for you, and that is that we have this pH indicator that actually contains all these other 4 indicators, and it gives you a gross idea of what the gross pH of the soil is. So what happens is you put the little soil in the cup, you add the duplex solution, and you turn it, stir it around, and you end up with sort of a color of the fluid that will match one of the colors on this card. For example, the first soil, 1A and 1B, both of those resulted in a pH somewhere between 4 and 5. The last 2 soils had somewhere in this greenish color with this kind of all-purpose pH indicator. Okay, so now that we know the range in the pH that we're interested in, we're going to now fine-tune the pH for that particular soil. And if you refer to the table on the previous page, to the table you're filling out, there's a little chart that talks about these 4 different pH solutions. And for example, the first one is called brome cresol green, and that's used if your pH range between 4 and 5. And for each one of these solutions, there's another little card that goes with that solution that has a range in colors that can more finely tune the pH in that particular range. For example, for the brome cresol green, it goes from about 3.8 down to 5.4. So that's one that you would use for the first sample that the samples 1A and 1B. And I'll show you how you're actually going to do this analysis next. I'm going to do, for an example, a soil 1B. And what you're going to do is just take a small scoop of the soil and put it in your little plate in the right hole so that you don't get these mixed up. And you don't need very much. And we're going to then take the indicator, the appropriate indicator for this pH range is the brome cresol green. And it has a range between 3.8 and 5.4. And we're just going to put in a couple of drops into the plate. 3 drops, I guess. And you'll notice that it's kind of a bluish green when you start off, but as we stir it, you want to stir it maybe, 30 seconds or so. Just make sure it's well mixed and it has a chance to sit. And sometimes with the colored soils that are kind of rusty red or yellowish like this one, it's a little hard to actually see the true color. So what you want to do is kind of look at the very edges of the plate, or edges of the drops here in the center of the plate, because that doesn't have much soil in it, and you can kind of get an idea of the true colors. Okay, then you're going to take the appropriate card for that particular pH indicator, and you're going to try to match it up as close as you can with the color of the solution itself. And in this case, we definitely aren't in the 3.8 range, and we definitely aren't in the 5.4 range. That's too blue. We're somewhere in between those two, and this is where it gets just a little bit tricky, especially for a colored soil. So what you want to try to do is kind of match the hue. Is it kind of more of a yellowish green, like this 4.2? Is it more bluish green, like this 4.6? Or is it in fact more like this 4.4 color over here? And I would say, just looking at this, that it's somewhere, I think, between the 4.4 and 4.6 in this case. Now, it doesn't have to be exact. I mean, if you said 4.4 to 4.6 in your answer, that's perfectly correct, because this is not an exact science. And again, these are just kind of field test kits that you could improve on if you were measuring this in the laboratory. But in this case, I would say that it's somewhere between 4.4 and 4.6. And so you're going to do the rest of these soils yourself, and using the appropriate pH indicator and the appropriate cards for each one of them. Okay, now we're going to test the humus content in the same sample that I just measured the pH in. And the way we're going to do that is we have a tube here that we're going to take a scoop of the soil. In this case, we're going to use a big scoop. And we're going to put this solution in, which is really just a solvent that will extract the organic matter from this test kit, also from Lamont. We're going to pour that in, and we're going to shake it up and mix it for about 2 minutes. So let me do that first. All right, soil in the big scoop, and of course it always spills, but that's okay. And we're going to fill this up to about the line of 7 or 8, somewhere in that range. And you want to make sure that you've labeled the sample, or at least you can distinguish it. You're actually going to do this for 2 samples, sample 1A and 1B. We'll have the other ones already done for you, and I'll explain how you read them in a second. So now that you've shaken this for a couple of minutes, we're going to now run the material through a filter to filter out the soil, and the liquid is going to have, if there is any, leached organic matter. And so what you're going to do is make a little triangle out of the filter paper that we have for you and open it up so it makes a little cup, and you're going to kind of rest it in there. You're going to have to hold it with your fingers, but rest it into this bigger cup. Take the lid off, if I can do that one-handed, and pour the whole solution in, and it's going to slowly drip through this filter paper. It may take a minute or so, and in fact sometimes to start it you have to give the paper a little bit of a squeeze, and you'll see that it'll start to drip, and once it starts to drip it kind of goes on its own. So we're going to have to wait and let that drip through, But what we're going to do while we're waiting is that what we have here is actually a really high humus standard that's also provided in the test kit so that you can compare your results to something that has a lot of organic matter in it. And so this one, remember I mentioned that if it looks kind of amber colored or the color of beer or recycled beer, that's got a high humus content. And we're going to compare those with the ones that you're doing yourself. Okay, so now we've let the solution drip through the soil and is collecting down the bottom of this cup, and we're going to compare this color basically with the color of the humus standard. And you can see that it's much paler yellow, and so this has a much lower humus content than does the standard that we first did. Now you're going to measure two of these, sample 1A and 1B, and we'll have, because this takes a little bit of time, we're going to have the other ones at the front of the classroom for you to compare. And you'll see that there is a difference among all the samples. So we want you to basically say this would be something like relatively high. I believe this is going to end up being relatively low. There might be some samples that are even lower than this in color, more pale. But once you've looked at all of them, you'll have kind of a relative scale from high, medium to low, and you'll fill that in the last column of your table. So now that you have all the data filled out in your table, we've gone over the color, the texture, the pH, and the humus content. We are now going to use that information to answer some questions about some of the physical properties of these soils and which soils might have the highest permeability, which of the soils might have the highest cation exchange capacity. Those are the sorts of questions we're going to ask. We're also going to ask you some questions about the pH, like which soils might be the best if there was some acid rain, which could neutralize more acid things, and which might be bad soils for acid rain contamination. Okay, there's one last thing that we can actually still determine from the soils that you've looked at, and this is something to do with sort of the geologic history that's recorded in the soils. Remember again that we had soils from Site 1 and Site 2 that were both, the bedrock was both Watchung basalt, but one of the sites, Site 1, was glaciated. And if you actually look at the pebbles in the sites, you'll see that there might be a difference. And we're going to have you look at the soils to see if there is a difference. We have these two containers with some of the pebbles and cobbles that were associated with the soils, and we want you to just look at them and pick up a few of them, and you have to go back to trying to remember what the soils look like. But you'll see here's an example of a pebble in Site 1. And what's the first thing that you notice about these? Are they, look at things like, are they all the same? Are they different? Do they have the same amount of weathering? Then you're also going to look at all the pebbles in Site 2. Again, you can look at there's several different particles, and but once they're broken open, again, do they all look kind of the same and uniform, or are they different from one another? You could also look at how thick is the weathering rind, we call this. How thick is the part that's being altered? This one, it's relatively thick, and in this one, similar kind of rock, it turns out, it's relatively thin. Okay, we're going to use that information to answer a few more questions about the relative ages of these two soils. Okay, so now that you've looked at these two boxes of rocks, so to speak, we also have a little stratigraphic column that shows where these samples were collected, the samples actually that you actually measured, and where these pebbles came from. And again, we have two sites, the Site 1 that was glaciated and the Site 2 that was unglaciated. And just by looking at these profiles and the pebbles that you saw in the containers, we're going to ask a few questions about the relative ages of these two soils.